So today we'll be talking about the cell cycle. So first we're going to start with interphase. In the interphase, you have your cell. And the interphase will be the longest phase of any cell cycle because it comes with a lot of parts. So first we're going to start with our nucleus. And inside our nucleus we have our messy DNA. We're going to put two of those because they're sisters. Messy DNA also stands for chromatin. So then over here we're going to put our centrioles. Right now they're on the side. So in the interphase they have three goals. G1, which is growth. S, which stands for synthesis. But in this part we will be talking about DNA replications. And then the last one is G2, which is also for growth. Our next phase will be prophase. And if you don't remember, pro means no. So in this phase, there will be no nucleus. Well, it will be, it's going to disappear pretty soon. Also, in this stage, it has 4 little steps to them. So, because our nucleus is going to disappear, we are going to draw little dotted lines because it's disappearing. And inside of this nucleus will be the chromosomes. They are going to be connected by the centromeres. It's what keeps them connected. And remember, our centrioles, they are going to start to move towards the poles. And spindle fibers are going to start forming. Your spindle fibers are like the fingers, and the centrioles are like the wrist. So our first step was your nucleus is going to start to break down. Then your chromosomes, they're going to come in and they're going to start to coil. Because remember, they were messy like a pig pen at first, but now they're going to start to coil and become chromosomes. Then our centrioles, they're going to move to the poles like north and south. And then our spindle fibers are going to form, as you can see. Our next phase is metaphase. This phase is very different than prophase. In this phase, we have our sisters. And if you remember, our sisters are the DNA. And in this phase, the sisters will start to line up in the middle with each other. And then we're going to take our centrioles and they're going to connect to the centromeres. And slowly start to separate the sisters from each other. So our spindle fibers will attach to the centromeres and move the sisters to the middle. Next phase is anaphase. In this phase, the sisters will now start to separate from each other and move towards the poles. This is called the sister split as well. Our last but not least is telophase. In this phase, our cells will start to split from each other. And it will form something called cleavage, which is right here. So in this phase, we will start to begin to create, well, not begin, but form two nuclei, as you can see. And in this nuclei, they will form messy DNA, again like chromatin. But another way to remember telophase is that it is the opposite of prophase. So, in this phase, our centrioles will no longer be at the poles, and they will each move into the two cells. And like I said before, our DNA will start to become messy again, and it will start to uncoil and become chromatin. And lastly, our spindle fibers will start to disappear from the cell cycle. Our last phase is cytokinesis. Oh, Jesus. Okay. So, in the cytokinesis, this is when our cells start to separate from each other. And like before, they both still have the messy DNA and the centrioles. And this phase is pretty easy to explain, because then this is the phase where the cycle will start all over again and do the same thing. So, this is called the cytokinesis phase, and it is very different from the plant cell. So, in the cytokinesis, unlike it starts to separate from each other, and the animal cell is more likely to separate from each other than the plant cell. The plant cell is more likely to separate from each other because it has this cell form that is around it to protect it from pinching and becoming two. So that is the big difference between the two. Thank you.